All right, hello everyone. We are going to cover our very last chapter today. It's over the nervous system, more specifically our special senses, like our touch, smell, taste, hearing, etc. All right. So again, roots, prefixes, and suffixes that you'll have to know as we go throughout this chapter. All right. So. Our senses, what do they do? Again, everything goes back to maintaining homeostasis. Um, our senses take information typically from the outside world as well as the internal environment. Outside world, we hear things from the outside world. We taste things that come in from the outside world. We smell things from the outside world, but and we touch things from the outside world. But we also have senses that look at what's going on inside our internal environment. We have pain receptors that are inside of our body that look at visceral pain. If you have a stomach ache, if you are having appendicitis, you might be feeling pain in your appendix area. So we can kind of classify those senses into two types of senses: general senses and special senses. General senses are those receptors that are located throughout the body. Special senses are structures that are really confined only to the head. Um, your special senses are eyes, ears, nose, and mouth. And typically, those are going to process information from the outside world, whereas general senses are going to process information either internally or from the outside world. Um, so typically, your various organs and joints are going to process information from the internal environment and the outside, or various organs typically are going to be from the internal environment. Joints can process information from the internal environment. You dislocate your knee, so you're going to have pain inside your body. Um, Skin is going to be kind of one of those that kind of fits both internal and outside world. You step on a tack, that's going to be the outside world. Or if you cut yourself, that could be both externally and internally. The cut itself would be externally, but the way that we um, kind of process the healing process would be more internally. As we know from the nervous system, those sensory receptors, those afferent pathways, are going to take that information and to the brain, spinal cord and brain. Then they link up with those interneurons, go to the brain, spinal cord, where the brain and spinal cord is going to process that information. And then, we have those motor neurons or those efferent pathways that are going to react. If you step on a tack, you're going to move your foot off the tack, or if you touch a hot stove, your motor neurons are going to remove your hand from the hot stove. But when we talk about our special senses, we actually have specialized cells that are going to deal with our special senses. All right, so we talked about the action potential and the receptor information, the receptor potential back in chapter 9 a little bit, when we talked about skeletal muscle contraction, and in chapter 10 when we talked about the -70, -55, up to +30 and everything. And our special senses kind of work in the same way. Um, but we're going to kind of put more definitions onto what actually is happening. We still have a threshold. We still have, depolarization, repolarization. But you can kind of see here that we don't necessarily have the typical um, graph of what we do for depolarization and repolarization that we saw in chapter 10 um, with our special senses. So what happens is that we have this stimulus that is going to stimulate our sensory receptors. Remember, the sensory receptors are going to be more of our eyes, ears, nose, and mouth. And then that stimulus is then converted into what's called a graded receptor potential, which is called transduction. And those potentials actually trigger those action potentials. So here's kind of what we see during skeletal muscle contraction with the depolarization and repolarization. And then those action potentials go through the neurons and stimulate the nerves and go to the sensory neurons into the central nervous system by a process called transmission. Now we talked about this in chapter 10 that the receptor potential, it's called a local graded potential that reflects the strength of the stimulus, in that you can differentiate me whispering versus shouting. You can differentiate something that is lukewarm versus something that is boiling hot. You can differentiate something that is sweet versus sour. And that's what that statement means. Just that you can reflect the strength of the stimulus. And that's why we have this kind of weird looking graph here, because we have different strengths of the stimulus. And then, of course, as we know, everything goes to the brain or spinal cord or the central nervous system. But when we talk about our special senses, actually we'll see that everything goes to the brain. All right, so sensory receptors. And I guess let me back up. Interpretation occurs to the brain. I said brain. That's not always going to be the case. If we're talking about eyes, ears, nose, and mouth, that's going to be going to the brain. If we're looking at our special senses, some of the skin, some of our pain pathways will go through the spinal cord. All right, so our sensory receptors. They're going to respond to specific stimuli. Obviously, your hearing receptors don't detect taste. Your vision receptors don't detect painful stimuli. That's a gimme statement. And it just allows us to interpret sensory events. They're very particular to certain type of environmental change and less sensitive to other stimuli. We'll talk about this when we talk about pain. We'll learn that pain is very sensitive. We don't adapt very well to painful stimuli, but our sense of smell, if we're repeatedly stimulated to the same sense of smell we kind of get used to that smell. You know, you go into grandma's house and she's baking an apple pie. If you spend, 20 minutes in the kitchen, you're going to get sensitive, you're going to get sensitized to that sense of smell, and then you leave the kitchen, you go outside for 10 minutes, you come back, and then you get that waft of smell again. So you get desensitized to that stimuli. All right, so what kind of receptors do we have? We have chemoreceptors, changes in chemicals, or looking at chemicals. We actually smell chemicals, we taste chemicals. Inside of our body, we are looking at our oxygen concentration. Oxygen is a chemical. If we're looking at our thyroid concentration, thyroxine is a chemical. Triiodine is a chemical. Blood glucose level is a chemical. So, there's an example of, external versus internal environment. So anything that we taste or smell is going to be a chemical. Pretty much anything that goes inside of our body, not goes inside of our body, but that is being detected inside of our body on an unconscious level through our autonomic nervous system, that you would get like on a blood report from your doctor's office, is going to be a chemoreceptor. Now, pain receptors, we actually call those nociceptors. So your pain receptors respond to either pain or tissue damage. So you cut yourself, that's going to be a nociceptor. So outside of pain, mechanical injury, electrical energy, you stick your finger in a light socket, don't do it. Thermal energy. And I said before, pain receptors or your nociceptors adapt very little, if at all. We don't want our bodies to become immune to pain. We still want those pain receptors to be active. Thermoreceptors. Thermo means heat. So thermoreceptors are going to detect changes in temperature. Now here I said moderate changes in temperature. We'll go over that, kind of what that word moderate means later on. Mechanoreceptors. Those are going to respond to mechanical forces. So think of like kind of pressure-based forces. So baroreceptors, it's a type of mechanoreceptor that's going to respond to pressure changes. So we have carotid baroreceptors. We have aortic baroreceptors in your aorta, in your carotid arteries, in your arterial walls. We have some baroreceptors, and that's going to adjust based on what it's going to adjust your heart rate, your blood vessel diameter, your arterial wall diameter, and it's going to adjust your blood volume based on what your blood pressure is doing. If your blood pressure is too high, you're going to vasodilate a little bit. When you vasodilate, your blood pressure is going to go down. If you vasodilate and your blood pressure is still too high, your body's going to tell your kidneys to get rid of some blood volume. So you're going to pee out a little bit of water. Also, these mechanoreceptors look at touch, tension, blood pressure, stretch. Remember that we have those Pacinian corpuscles and those Meissner's corpuscles, those tactile corpuscles that are touch. Those Pacinian corpuscles that look at pressure, that are in our dermal layer. Photoreceptors. Photo means light, so your photoreceptor is going to be pretty much in your eyes only, that are going to respond to light. All right, so sensory neurons, or sensory receptors, are going to be at the ends of neurons. Remember those neurons have dendrites, axons, or dendrites, cell body, and axons. So those sensory receptors are kind of specialized dendrites. And you know, as those receptors pick up that information, they're going to cause that local change in that membrane potential, just like we saw with skeletal muscle contraction. And it's going to cause that, that graded potential according to that stimulus intensity. And remember that, you know, the peripheral nervous system is going to transmit the impulses to the central nervous system via the afferent pathway, where they're going to be analyzed and interpreted in the brain. All right, so we have kind of three definitions: sensation, perception, and projection. And they kind of fall into into progressively more detailed order. So sensation is the least detailed order, and projection is the most detailed order. So sensation is basically, you know something's going on, but you're not sure exactly what it is or where it is. So, So here the example, so basically it occurs when action potentials make the brain aware of something. So here the example is, you know, your retinal cones detect color, and it's going to send impulses to your visual cortex. So basically you're, in your vision, we'll talk about rods and cones. Cones detect color, which we'll get into later on. But so your eyes see something maybe in your periphery. Or another example, you're aware of pain, but you really haven't isolated where that pain is. And then perception is where your brain has really become aware of that pain. Your brain is interpreting that pain. So here, you know, that pain is a result of stepping on a tack. Or going back to that rods and cones example, now you're aware of that person in that periphery is wearing a red coat. And then projection is where your cerebral cortex interprets that sensation. So, you know, sensation, something's going on in your periphery. Perception, that person's wearing a red coat. Now projection is, you can say that person's wearing a red coat. They just walked out the door and they're 30 feet away. Now you are aware of the location where that stimulus occurred. That person is no longer in your periphery. You've turned your head and you can see that person's 30 feet away. It's a female with blonde hair, and they are wearing a red coat and they're about 30 feet away. So you're fully aware of that stimulus. That's called projection. Going back to the stepping on the tack example, you are fully aware that you've stepped on a tack. You've done your withdrawal reflex, your crossed extensor reflex that we talked about a couple chapters ago. You're now cussing because you stepped on a tack and oh my gosh, it hurts so bad. You are now in projection. You are fully aware that you've stepped on a tack. You're yelling at your dog to get out of the way because now your dog's barking. Anyway, so sensation, something's going on. Projection, you're kind of aware that what is happening. Projection, you're fully aware of what's going on. All right, so with our different special senses, smell, taste, sight, and hearing, we have kind of like a flow of information of how things are going to come about. And you're going to need to know a little bit of information about how this information flows. So first of all, we have our sensory receptors that are going to pick up the information. And somewhere we're going to have that impulse in that sensory fibers. So those sensory fibers pick up that impulse. And then that impulse is going to reach the central nervous system. And then we have a sensation. It could be a new sensation, it could be a recalled memory. And then we have perception. We're not going to get into that projection, last one in this information flow. So when we look at smell, our sensory receptors for smell is our olfactory receptor cells. And we'll look at this when we talk about olfaction, sense of smell. So our sense of smell is our olfactory receptor cells. And that goes through, if you remember, the cranial nerves. That's our olfactory nerve fibers. Do you remember what cranial nerve the olfactory nerve is? We just did it. It should be cranial nerve number 1. All right, so cranial nerve number 1, your olfactory nerve, then goes to the cerebral cortex. Remember, we have to go to the central nervous system. And then that sensation would be a pleasant smell. And then what we're going to perceive is the smell of an apple or the smell of an apple pie or whatever. For taste, we have taste bud receptor cells. And then we have those sensory fibers. We have a bunch of different cranial nerves that are involved in the sense of taste, which is going to go to the cerebral cortex. We're going to taste something sweet. It could be the taste of an apple. For sight, remember, we're going to have rods and cones that are located in our retina. The optic nerve picks up processes our visual information. Do you remember what cranial nerve our optic nerve is? It's cranial nerve number 2. Our optic nerve then goes to the midbrain and the cerebral cortex, and it's going to process that information as a small round object. Remember, that's our sensation. If we go back to what our definition of sensation is, we're aware that something is going on, but we don't necessarily know what it is. A small round object, and then our brain processes that information, and now it processes it as an apple. In hearing, we have these things called hair cells that are located in this structure called the cochlea or cochlea, however you want to pronounce it. And then we have these auditory nerve fibers. You're going to need to know what cranial nerve the auditory nerve is. And then it's going to get processed in the midbrain cerebral cortex. We're going to process it as a crunching sound, and then the brain will process it as a sound of biting into an apple. So just kind of know a little bit about the information flow. All right, so here is a question. After taking a bite of pizza, sensory receptors are stimulated in your mouth. What is the order of information flow that results? So we know that we stimulate our sensory receptors. So far A, B, C, and D are correct. And then if we go back to this page, basically we have sensation and perception are the last 2. So we can cross off this one. We can cross off B because perception should be the very last thing, so that one, those 2 would be out. So here perception is the last thing. And then, so from here we stimulate, then we send information to the central nervous system. Then we go through perception or sensation and then perception. So B and C are out. Stimulation, and then here these two are flip-flopped. So D is out. So A would be our correct answer. Stimulation of sensory receptors. Stimulation of sensory receptors. Impulse sent to CNS. There's that one. Then we have sensation and perception. Sensation and perception. All right. So sensory adaptation. Remember I said that sometimes we get used to things. It's the ability to ignore unimportant or continuous stimuli. So the ability to ignore unimportant stimuli. So we see a lot of things every day, but we don't necessarily recognize everything that we see. Our brain is good at kind of filtering out a lot of things. We hear a lot of things, but we don't necessarily register everything that we hear. Our brain kind of ignores a lot of unimportant conversations, a lot of unimportant things that we see that we really don't process. And then it says ability to ignore continuous stimuli. And I use that example about, you go in, you smell the apple pie, and then you leave, and then you come back, and you kind of get sensitized to this stimuli. So, once you go in the kitchen and you smell the apple pie for 20 minutes, it doesn't smell as pungent or as strong as it did when you first walk into the kitchen, as it did, 20 minutes ago. And that's because of the of sensory adaptation. And the example that the book uses is that the smell of vomit fades as the time goes on. And just remember this statement for when we get to the sense of smell, because I'll come back to this, and I think it's funny. All right, so basically the sensory impulses become less frequent, and they actually may cease. So you could be standing in the kitchen and you might not even smell the apple pie anymore. And going on with what the book says, you might not smell the vomit, even though you're standing in the room full of vomit, which I would probably disagree with, because I can promise you I'll still be smelling vomit. And then because of that sensory adaptation, we're going to need a stronger stimulus would be required in order to trigger that impulse. And I look at this as let's say that you're taking opiates to treat your pain. You need more and more and more pain medication if you become addicted to those pain meds to treat that pain. You're going to need more and more. So you're going to need a stronger medication is required to treat that pain. That's how I look at that. Thermoreceptors, so your heat receptors and your moderate heat receptors and your olfactory, your nose, your smell receptors, are what adapt the most. All right, so we can divide these receptors that are in our skin, muscles, joints, and kind of inside of our body, those general senses, into three different groups. Those exteroceptive senses, interoceptive senses, and proprioceptive senses. So exteroceptive senses are basically associated with just the body surface. Touch, pressure, temperature, pain. Remember, those are tactile corpuscles, our Pacinian corpuscles, our thermoreceptors, our nociceptors. Our interoceptive senses, we also call them our visceroceptive senses. Remember, viscero is kind of like our organs inside of our body. That's our autonomic nervous system. So those are senses associated with changes inside of our body, inside of our viscera. Um, Blood pressure, stretching our blood vessels. Remember, we have that elastic tissue, or those that elastin connected to our arterial walls that help vasodilate and vasoconstrict to accommodate blood volume changes, blood pressure changes. Proprioceptive senses, remember those are called proprioceptors. We can also say proprioception, and those are senses associated with changes in tension in muscles and in tendons, in joints in our body. So those are going to be stimulated when changing position or exercise. So proprioception is another way of saying more tension. All right. So when we look at our mechanoreceptors, we can kind of have three types of mechanoreceptors, and typically these are going to be exteroreceptors. Remember, exteroreceptors or exteroreceptive senses are, touch, pressure, temperature, pain. These are going to be free nerve endings. They're very common in our epithelial tissues. Remember that dermal layer. They're going to be very simple in their structure. Itching, other sensation. We talked about those Meissner corpuscles, those tactile corpuscles, detect touch, texture, very abundant in our dermal layers, especially in our hairless portion of our skin and lips. Those lamellated or those Pacinian corpuscles that detect pressure, stretch, vibration. Those are found more deep in that dermal layer, as well as our subcutaneous tissue, that hypodermal layer, and as well as our tendons and our ligaments. If you remember, we looked at kind of this slide a little bit in our, chapter 5 or something. In our dermal layer, there's going to be our lamellated Pacinian corpuscles. There's going to be our tactile corpuscles. We have free nerve endings. Those are all exteroreceptors. All right, so, way back up in this slide, I said thermoreceptors respond to moderate changes in temperature. So thermoreceptors, they're also free nerve endings in skin. So when I say moderate changes in temperature, we have warm and cold receptors. So basically, we're looking at, without reading the slide, basically it says sensitive to 77, and then our cold receptors basically work between 50 and 68 degrees, our cool temperatures, and then our warm temperatures between 77 and 113. So between 77 and 113 are our warm receptors, and between 50 and 68 are our cool receptors. So pretty much anything above 113 degrees, we're not working off of our thermoreceptors, and anything below 50 degrees, we're not working off of our thermoreceptors. Anything below 50 degrees, we're actually switching over to our pain receptors, our nociceptors. And so if you're outside shoveling snow and if you're feeling that cold, you're actually working off of your pain receptors, your nociceptors. If you're living out in Arizona and you're really, really, really hot, you might be working off of your pain receptors. Um, if it's greater than 113 degrees, typically here in North Carolina we don't get above 113 degrees outside, but some places in the country it does. All right, so what is the destination of fibers of the spinothalamic tract transmitting pain and temperature information? All right. So this one should be an easy one. Don't read into the question. Remember several chapters ago we talked about ascending and descending tracts in the spinal cord, and I taught you how to name them. This is an ascending tract. It's going from the spine to the thalamus. So B is going to be our answer. We had the rubrospinal tract. So it's going to go from somewhere in the brain to the spine. We had the spinothalamic tract. We had a lot of different tracts that we named. Basically, if there's a part of the brain going to the spine, it will end with spine. If it's going from the spine to the brain, it's going to start with spino, and then it will end with some structure in the brain. All right, so our pain receptors, we call those nociceptors, basically a bunch of free nerve endings widely distributed throughout the body. Basically in the brain or in nervous tissue of the brain, we don't have any pain receptors. So, I could cut open your skull and I could actually poke in your brain and you wouldn't be able to feel it. You wouldn't be able to feel the pain because there's no pain receptors there. So those nociceptors stimulated by tissue damage. If you cut yourself, you're going to feel pain. If you step on a tack, you're going to feel pain. Chemicals, mechanical forces, or extremes in temperature. We talked about less than 50 degrees, greater than 113 degrees. Oxygen deficiency. If your oxygen becomes too low, you will actually feel pain. Adapt very little, if at all. We talked about sensitization, and pain does not, pain receptors do not become sensitized. All right, treatment of pain. Typically we're going to go for the NSAIDs, ibuprofen, aspirin, are going to be your first line of defense. However, they can cause upset stomach. Opioids, we talked about opiates before, they can be addicting. Acetaminophen, that's Tylenol, does not really cause any gastrointestinal irritation, but because Tylenol or acetaminophen is processed by the liver, it can damage the liver if you take too much Tylenol, or if you take Tylenol chronically. So people who try and commit suicide or overdose on Tylenol, really you're not going to kill yourself by Tylenol, but you're just going to put yourself into liver failure. People who try and kill themselves with an overdose of aspirin, you're just going to blow out your kidneys. Chronic pain. There's a lot of people out there with chronic pain treatment, with chronic pain. There's complex regional pain syndrome, type 1, type 2. How do we treat chronic pain? Again, NSAIDs, yes, we do treat chronic pain with opiates. Exercise, injections, we can do nerve blocks, we can do epidural steroid injections, antidepressants work. We're finding that a lot of antidepressants like duloxetine actually helps with pain management. TENS unit, I already talked about nerve blocks. All right, so visceral pain. Remember, visceral is kind of inside the body. We have pain receptors that are inside of our body. However, we don't necessarily experience visceral pain the same way that we do with pain on the outside of our body. If you remember from several chapters ago, we had those dermatomes, and we had a slide picture of those dermatomes, and we had like a bunch of different lines on the body, and we could say that if I had, pain sensation in my nipple area, that could be traced back to a specific spinal nerve. If we had pain in our calf muscle, that could be traced back to a specific spinal nerve. If we felt sensation in our, pinky finger, that could be traced back to a specific spinal nerve, one of our cervical nerves. When we're dealing with pain inside the body, it's not necessarily the same thing. We call this referred pain only because we don't have those same types of dermatomes inside the body. If you've ever heard of like someone having a heart attack, that they feel like, the elephant sitting on the chest, or that they have radiating pain down their left arm. Obviously your heart is not in your left arm or in their left shoulder. That is referred pain. Because those impulses arise from that visceral organ, they could synapse with different neurons within the central nervous system, so it might feel like that the pain is originating elsewhere. So if we look at what's going on inside the body, here, if you're having heart pain, you might feel that heart pain going all the way down your arm, elephant sitting on your chest. You might have some pain in your back, just because that's how the nerves kind of link up in the central nervous system. If you're having kidney pain, your kidneys actually sit about right here in your back. But look at where you can feel your kidney pain. Your bladder actually sits right here, but you could actually feel bladder pain all the way down, kind of like the top parts of your legs, on the medial aspect of your legs. So that's actually referred pain, because the pain does not necessarily lie where the organ actually is. And it's just because those nerves that are innervating the organ link up with other nerves in the central nervous system. All right, so when we talk about nerves, we have different types of nerves. There's other different types of nerves than the ones that are listed here. And there's subtypes of fast nerves, and there's subtypes of slow fibers. But we're only going to talk about kind of like the two big ones, fast pain, A delta fibers, and slow pain, which are C fibers. And like I said, there's many more different types out there. Remember that anytime we see fast, we should think of myelination. Remember that lipid protein coating that's on the axon of the nerves that leads to a faster action potential. Typically, this is going to be associated with sharp pain or acute pain that is very localized, very more pinpoint into an isolated area. Once that stimulus stops, usually the pain is going to go away. Slow pain fibers, which are type C fibers, typically those are going to be unmyelinated, no myelination, so those are going to be slower. That's going to be more associated with a dull, aching pain or more of a chronic pain. More difficult to isolate or localize, and typically that pain is going to continue after the stimulus stops. Now with our pain pathways, remember that those pain pathways go up the lateral spinothalamic tract, up through the reticular formation, and then goes through that thalamus, spinothalamic tract, and then into the hypothalamus and cerebral cortex. All right, so you drop a book on your foot, immediately feel a sharp, stinging pain. What type of pain fibers are responsible? Sharp, stinging pain. So sharp pain, immediate, fast pain fibers. Dull, aching pain, slow. All right, pain pathways. Remember, spinothalamic tract. So that is going to go up into the thalamus, where you're going to have that sensation of pain. And then it's going to get processed by the cerebral cortex, where it's going to have the cerebral cortex is going to look at the intensity of pain, where is that source of pain. That's where you're going to start cussing and yelling. It's going to provide the motor response to the pain. You're going to start jumping up and down. And then that emotional response is going to be because of that limbic system. Remember, the limbic system does a lot of our emotional processing. All right, remember our body has natural pain inhibiting substances. Remember those enkephalins, endorphins. Enkephalins are basically our body's natural Tylenol and Advil. Serotonin releases or stimulates the release of enkephalins. We also have endorphins that kind of gives our body a natural high. Mechanoreceptors. Remember what mechanoreceptors do. They look at the tension of skeletal muscle and tendons. So they're going to help, gymnasts kind of make sure where they are in the air, help pole vaulters know where they are when they're doing their pole vaulting. We already talked about proprioceptors or proprioception, those lamellated Pacinian corpuscles. Remember Pacinian, pressure. We also have muscle spindles. Muscle spindles are kind of stretch receptors in skeletal muscles. These muscle spindles basically will initiate this thing called a stretch reflex, which if you overstretch a muscle, it will actually kind of cause a muscle contraction. So if you're overstretching a muscle, these muscle spindles will actually contract the muscle so that you don't kind of rip your muscle. We also have these Golgi tendon organs that are actually in the tendons, hence the name Golgi tendon organs, that will be activated once a muscle is kind of stretched to its limit. So the muscle spindles will actually work on the muscle, but these Golgi tendons will actually work on the tendon, and they also stimulate a reflex that opposes those stretch reflex, and they help maintain posture and provide muscle attachments. Um, so this is just a picture of that, these Golgi tendon things, and these muscle spindles. So these muscle spindles will actually cause a muscle contract if they are being stretched too much, and these Golgi tendon organs will cause uh, the um, once a muscle is nearly stretched to the limit, it will help stimulate that reflex, to say, hey, I don't want my muscle to tear, so let's go ahead and contract a little bit. All right, so you're carrying a box when your friend suddenly places another box on top of it. Your muscles respond by immediately contracting. What receptor senses the rapid muscle lengthening and causes a reflexive response? All right, so here we're talking about what receptor senses the rapid muscle lengthening and causes, there we go. What receptor sensed the rapid muscle lengthening and caused a reflexive response? So, so we can get rid of fast pain fibers. We can get rid of tactile Meissner's corpuscles, because remember, tactile deals with touch. Fast pain fibers really is pain. There's nothing in here that talks about pain. Muscle spindles or Golgi tendon organs. So muscle spindles deals with muscle lengthening and contraction. Golgi tendon organs deal with tendons. So basically this question is asking about muscle lengthening, so we're going to go with muscle spindles. All right, so visceral senses. Remember, visceral, internal organs. They're going to have lamellated corpuscles, a bunch of free nerve endings. Basically, these are going to let us know when we're full. If we have, a bellyache, if we have, if we need to burp. Oh, my phone's ringing. All right, burping. Actually, I just learned something. I didn't know this until the other day. Did you know that burping has a medical name? Eructation. Eructation. That's another name for burping. There is your useless fact for the day. Anyway, also signals that you're having a heart attack. All right, so just a summary slide of different types of receptors that we have, what they do. Just be familiar with this slide. Special senses, remember sight, sound, taste, hearing, sense of smell, olfaction, that's going to be in our nasal cavity. Taste, taste buds are in our oral cavity. Hearing, in our inner ears. Actually, hearing, our inner ears not only does hearing, but our equilibrium. You get dizzy on a roller coaster or sitting in the back seat of a car. Sight is with our eyes. So, synesthesia is basically when your senses kind of get mixed up. There's it's a condition where your brain interprets a stimulus from one sense coming from another. So, an example here, the paint smelled blue. Obviously, a paint can't smell like a color. It's actually caused by a genetic mutation. And these people actually are usually higher on the uh, smart scale. Um, they're very common in creative people. Usually these people are going to be more artistic. Um, and a lot of these people kind of compartmentalize these things, and they see a lot in color. Not that they see like auras or um, but they can some of these people can like tell you the license plate of the last 100 cars that went past their house, or they could tell you what they had for lunch on, you know, July 27th, 1982, if they were alive back then. Anyway, all right, so with olfaction, your sense of smell. Um, Sense of smell, we smell chemicals. Those chemicals are actually dissolved in liquids. Um, you know, essentially your snot. The mucus that is produced by your nasal cavity. Your sense of smell and your sense of taste are very much related. About 75 to 80% of your sense of smell is related to your sense of taste. We have these receptor cells, olfactory receptor cells, where they're actually bipolar neurons. Remember bipolar neurons, we have our dendrites that come in, and we have one axon that leaves. So contains, you know, upper parts, nasal cavity, the superior nasal concha. We'll look at your nasal septum. Your nasal septum is what you get pierced if you have a nose ring, like that circular ring that hangs out of your nose. We can smell almost 400 types of smells. So we have about 400 types of membrane receptors. So here you can see your nasal cavity. We have a superior, inferior nasal concha here. And then this is our olfactory area of the nasal cavity. That's actually called your cribriform plate. And if we blow up that area, you can see the cribriform plate here. We have, remember, our columnar epithelial cells. And then we have our bipolar neurons that are going to have a bunch of cilia on the columnar cells. They're going to remember that cilia is there to um, increase area for absorption, as well as to move things through the nasal cavity. Remember mucus production, respiratory tract. We have the receptor cells, and then everything is going to link up with the olfactory nerve, cranial nerve number 1. All right, so remember that slide that we looked at earlier that talked about the apple and the information processing. So once the olfactory receptors are stimulated, those nerve impulses are going to travel through openings in that cribriform plate. This cribriform plate is actually in this bone called the ethmoid bone. And then that olfactory nerve then goes through the olfactory bulb. So if we go back, let me go back to this. Here what we're doing is we're actually kind of expanding this area right here. So we're talking about the olfactory nerve. And then it's going to pass through these things called the olfactory bulbs, then the olfactory tract, then to the limbic system. Remember, we're going to have an emotional response. If you, let's say you smell your older brother's fart, you might, and then the olfactory cortex for interpretation. And then, sensation, projection, and those olfactory bulb processes sensory impulses. Remember that limbic system is for emotion, provides emotional responses to that any type of odorant molecules, whether it be your fart smell or the apple pie that reminds you of grandma and might makes you smile. All right, so olfactory stimulation. So remember I told you that we can smell about 400 types of smells. So each olfactory receptor, we have about 400 types of them, can smell one type of membrane protein. And then each protein combines several types of odorants. And it can undergo sensation adaptation rapidly. Remember, thermoreceptors and olfactory receptors can undergo adaptation rapidly. Now, remember I said that we had that smell of vomit and that it can fade rather quickly. It says here the sense of smell drops by 50% within 1 second after stimulation. And I said that, we'd come back to the vomit smell comment. I can promise you that if I'm in a room full of vomit, it does not drop by 50% within 1 second after stimulation. But that's what the textbook says, so we're going to go with it. And if we look at, this slide, remember we're breathing in air. So these neurons right here really are the only receptor neurons that come in direct contact with the outside world. So you could actually damage these neurons. If you pick your nose and if you stick your finger all the way up your nose, you could actually feel those neurons. And they're actually replaced rather regularly. Remember, typically neurons don't get replaced. But in the olfactory system or in the olfactory senses, they are. So there's a lot of research being developed as how are these being replaced if it's very hard for neurons to regenerate. All right, taste buds. We obviously, taste buds are our organs of taste. They're located on the papilla of the tongue. You actually have taste buds on the roof of your mouth, on your cheeks, and in the walls of your pharynx, kind of in the back of your throat. We have about 10,000 taste buds, and then each taste bud contains between 50 and 150 taste cells. So we actually have about 1 million taste cells in our mouth. 10,000 times 100. So our taste receptors, obviously we're tasting chemicals. Your taste cells are basically modified epithelial cells. You can see here there are a bunch of stratified squamous epithelial cells. And then you can see here that we have these hair cells, these taste hair cells. Basically, they're a bunch of microvilli. So kind of like nerve endings that that protrude through the taste buds. They're basically very sensitive. Um, and just like with a sense of smell, your sense of taste requires that molecules bind to these receptor proteins. Taste cells are actually replaced about every 3 days. We actually have about 5 taste sensations: sweet, sour, salty, bitter, and umami. Sweet is stimulated by carbohydrates. If you remember, carbohydrates get processed down into glucose, so that's why we taste sweet. Sour is stimulated by acids. Remember, acids are anything that can be dissociated in producing a hydrogen ion. Salty, stimulated by salts, anything that can contain sodium or potassium. You know, sodium chloride, potassium chloride. So sodium, potassium are the salty component. Bitter, stimulated by a lot of organic compounds such as magnesium, chloride, salts, strychnine, nicotine. And I thought strychnine was interesting because strychnine is actually a poison. Umami. Umami is stimulated by some amino acids in MSG. MSG is in a lot of Chinese food. And actually, this umami was actually just added to the list maybe about 20 years ago, 25 years ago. Before then, we only had the first 4 taste sensations, but they just kind of recently added umami. Here also, spicy foods can stimulate actually some of your pain receptors, some of your nociceptors, and spicy foods also stimulate some goblet cells to release some mucus. So your taste receptors undergo rapid adaptation. So for those of you who like spicy foods, as you eat spicy foods over and over, you become accustomed to eating spicy foods. All right, so here you have the protein miricurlin. It's kind of made up, which is found in these taste-altering miracle berries, causing acidic foods like citrus fruits, soda, and vinegar to taste like candy. Which of the following is the most likely mechanism for the effects of miricurlin? So basically, we're taking acidic foods to taste like candy. So miricurlin alters sweet receptors to perceive carbohydrates as sour. So basically, we're taking acidic foods to taste like candy. So we're looking for something to taste acidic to candy. So candy is sugar, and sugar is carbs. Sugar is also sweet. So let's look for those. So miricurlin alters sweet receptors on the tongue and causes them to perceive carbohydrates as sour. So that one's going to be out because we're altering acid and causes carbohydrates Nope. All right. Miricurlin alters sour receptors on the tongue and causes them to perceive acids as sweets. So that one so far looks good. Miracurin inactivates sweet receptors, so only sour receptors will detect sweet compounds. No, we're not actually altering no, we're not inactivating our sweet receptors because we're still perceiving sweetness. All right, miracurolin amplifies umami. No, this has nothing to do with umami. So the answer is going to be B. So we're altering our sour receptors on the tongue and we're causing them to perceive acids as sweets. So that's going to be our answer. All right, sensory impulses from the taste receptors on fibers 3. So, okay, taste pathways. Remember I said a bunch of cranial nerves involved in our taste sensation. Cranial nerve number 7, facial nerve, glossopharyngeal number 9, vagus nerve number 10. Remember, the vagus nerve is probably the most important nerve in the body. It innervates essentially every organ inside of our body. And then those nerve impulses are eventually going to go to the medulla oblongata, portion of the brainstem, then proceed to the thalamus, and then into that insula or that Isle of Rail. We have a gustatory cortex. Gustatory just means to taste. Now we can alter our sense of taste. If you have a cold, flu, allergies, will alter our sense of taste. Nasal polyps, remember our sense of taste and smell are very interrelated. 75 to 80% of our sense of taste and smell are interrelated. So anything that alters our sense of smell will alter our sense of taste. Nose injury, head injuries, chemical exposure, nutrition, medical problems, disease. Remember, COVID-19 was, knocked out our sense of smell. Some cases are temporary, others are permanent. Sometimes we can't identify a cause. Medications can actually alter the taste of smell, alter your sense of taste or sense of smell. Sometimes if you're given a medication, you might taste the medication. Like if you're given an IV medication, sometimes you can taste that medication as it goes throughout your body, throughout your bloodstream. All right, in the ear, we have the ear can be divided up into 3 sections. The inner ear, or the outer ear, the middle ear, and the inner ear. The ear is actually the organ for our hearing and equilibrium. The outer ear, we call it the external ear. That's the part of your ear that obviously you can see. So, the big portion of the ear, that's called the auricle or the pinna. You can see that it's divided up into a bunch of different structures. You don't have to memorize any of those. Basically, it's a funnel-shaped portion that's just going to collect all those sound waves and direct those sound waves into what's called the external acoustic meatus. That is, the portion of your ear that you stick the Q-tip in. It's an S-shaped tube. It's lined with ceruminous glands, and that produces what's your earwax. And it carries the sound to your tympanic membrane, which is your eardrum. And your eardrum is going to vibrate in response to those sound waves. And then once we get to the eardrum, that's going to be the kind of end of the outer ear. All right, so question. When cleaning his ear with a cotton swab, Q-tip, Garrett pushes the swab into the external acoustic meatus as far as he can. When he is met with resistance, he pushes harder and suddenly feels a sharp, intense pain. He then notices that all he can hear from that ear is constant ringing. What damage has he done? Most likely to his ear. He has torn the oval window. Um, so vibrations not being transmitted to his inner ear. He has damaged the stapes, he has torn the skin, or he has torn his tympanic membrane. So basically, we haven't talked about the oval window yet. We haven't talked about the stapes yet. We haven't. There's nothing in the question that says he's torn his ear canal or that he is bleeding. Basically, he's actually torn his tympanic membrane, which is preventing the tympanic membrane from vibrating and transmitting those sound waves to the middle ear. So that's going to be our answer. And so if you do kind of push the Q-tip too hard, you can rupture your tympanic membrane. Or if you are on a flight, or on the plane, you plug your nose and you kind of blow out a little bit to kind of equalize that pressure. You could actually rupture your tympanic membrane that way. All right, so continuing on, our middle ear, we have that tympanic cavity. So your outer ear obviously is a bunch of air-filled stuff because we have air on the outside. Your middle ear is an air-filled pocket as well inside the temporal bone. And we have 3 tiny bones that are located inside of the middle ear, called the malleus, incus, and stapes. And if you look at them, they look like a hammer, anvil, and a stirrup. So you can see how tiny they are. There's a penny. And those are going to vibrate in response to the vibration of the tympanic membrane. And basically all our job is to amplify that vibrational force. And then we have this structure called the oval window, which is going to be the beginning of the inner ear. Now the inner ear is a fluid-filled pocket, which we'll talk about once we get to the inner ear. But so here you can see, here's our tympanic membrane, or our eardrum, and the sound waves come in here, and it's going to vibrate. It's going to knock up against the tympanic membrane, which is going to vibrate, which is going to cause the malleus to vibrate. Then it's going to cause the incus to vibrate. Then it's going to cause the stapes to vibrate. So now we have the eardrum, the malleus, the incus, and stapes to vibrate. Now the stapes is actually connected to this oval window, which you really can't see because the stapes, like I said, is connected to the oval window. And as the stapes is kind of, the stapes then will kind of vibrate or beat up against that oval window. And remember, the inner ear is kind of filled with, is a fluid-filled pocket. So now we're taking kind of these sound waves, and now we're kind of throwing a stone in a pond, and we're kind of turning these sound waves into other types of sound waves, more fluid sound waves. And now we're making a ripple effect in more of a water-filled pocket. So now we're going to talk about this thing called a tympanic reflex. So basically, a tympanic reflex is going to be seen when we have loud sounds. When that malleus, incus, and stapes kind of vibrate, and we have a bunch of loud sounds, your body's going to try and protect itself. Your body will basically decrease those vibrations that are transmitted to the inner ear. Those vibrations are actually transmitted less effectively. We have muscles that are involved in vibrations of the malleus, incus, and stapes. It's not like those bones move automatically. So, this tensor tympani and the stapedius, actually will kind of not work as effectively or as efficiently as it typically does, because the body's trying to protect itself. It doesn't want those loud vibrations to be transmitted to the inner ear. And we'll talk about hearing loss related to loud noises or loud music later on. So right now, tympanic membrane vibrates, malleus, incus, stapes vibrates. That stapes butts up against that oval window and is vibrating against that oval window. Now, before we move on, we have this thing called an auditory tube that's down here. Remember, we have that external or that ear canal where you stick your Q-tip in, but we also have this auditory tube as well. It's also called the eustachian tube, and it connects the middle ear to the throat. So I use the example of, you're on a plane and you hold your nose and you blow out. Basically what you're doing is you're trying to equalize that pressure on both sides of that tympanic membrane. Remember, that middle ear is a air-filled pocket, the outside air is an air-filled pocket. So you're trying to equalize that pressure when you plug your nose on an airplane, and you're trying to equalize that pressure. There's usually a valve-like flap in your throat that you could open up when you plug your nose and kind of blow out and equalize that pressure. Now, changes in altitude might make you feel that that pressure inside of your head. And when you equalize that pressure, you can hear a popping sound. Also, I said it actually I didn't say. It connects that middle ear to the throat, so that nasopharynx of the throat. So if you use eardrops, they can actually go through that tympanic membrane. And you can sometimes taste the eardrops on the back of your throat. Remember, you have taste buds in your pharynx, and those eardrops, you actually place those eardrops here, and they can actually go into your middle ear and then travel down into the back of your throat through the eustachian tube or auditory tube. All right, so middle ear infections, we call that otitis media. Basically, it's treated with antibiotics. A lot of young children have this because those auditory tubes, that eustachian tube, is very short. You can get a sore throat. A lot of bacteria can get in your throat and migrate backwards up into your middle ear. And if you have a lot of ear infections as a kid, we can put tubes in your ear. It's actually about a maybe 10-minute anesthetic procedure, surgical procedure, and they usually last for about 6 months to a year before they'll just fall out. All right. All right, the inner ear. Remember, the inner ear is a fluid-filled pocket, and it says consists of labyrinths located in the temporal bone. Labyrinths kind of just means a maze. So we have this bony labyrinth, or this osseous labyrinth. So it's a bony canal inside the temporal bone with this fluid called perilymph. And then we also have a membranous labyrinth filled with fluid called endolymph. Now, we do have three portions of this maze or this labyrinth. We have this cochlea or cochlea, that is our hearing organ. We have these semicircular canals that are going to help with dynamic equilibrium. And then we have these vestibule things that are going to help with static equilibrium. So if we're looking at this picture, kind of here's a picture of our inner ear. So we're going to start with this oval window right here. This is what the stapes butts up against when it's vibrating. Then we have these this organ right here called the cochlea, which is what we hear with. Then we have these semicircular canals that are involved with dynamic equilibrium, which we'll talk about later. And then we have these this section right here, which are the vestibules, which are involved with static equilibrium, which we'll talk about later. So there's three portions to the inner ear: your cochlea, hearing, your semicircular canals, which are dynamic equilibrium, and your vestibules, which are static equilibrium. Remember, perilymph is within the canals, so like your semicircular canals, and your endolymph is within those ducts. All right, so I mentioned that we have that oval window that the stapes is kind of hitting when it's vibrating. Now, as the stapes is pounding against the oval window, we also have this structure called, so here's the oval window. We also have this structure called the round window. So as the stapes is hitting the round window, we have this round window, or as the stapes hits the oval window, the round window will kind of jet out to help relieve that pressure. Because remember, the inner ear is a fluid-filled environment. So the oval window pushes in, the round window will jet out to help relieve some of that pressure. So that round window will dissipate excess vibrations into that tympanic cavity, into that inner ear. All right, so the cochlea. Remember, that's our hearing, our main hearing organ. It kind of looks like a snail-shaped tube. So here you can see the cochlea, kind of looks like a snail. It's coiled around this bony core called the modiolus. And there's three compartments to the cochlea: this scala vestibuli, scala tympani, and this cochlear duct. Now we're really only going to talk about this cochlear duct. Basically, this cochlear duct has this is the receptor organ for hearing. So even though I said that the cochlea is our hearing organ, the receptor organ for hearing is called the spiral organ, or the organ of Corti. And this is the one that you're going to have to know for exam purposes. So the cochlear duct contains the receptor organ for the organ for hearing, and that's called the spiral organ, or the organ of Corti. So here, if we kind of stretch out or unwind this snail shell and make it into a straight line, you can see this cochlear duct right here, and inside that cochlear duct, that's where we're going to find all of those receptors for hearing. You know, back here I said the scala vestibuli and the scala tympani. There's your scala tympani, scalar vestibuli. You're not going to really need to know that. But the one that you need to know is that cochlear duct. It's going to have that endolymph in it, but it also contains those receptors for hearing. It's called the organ of Corti, or that spiral organ. And we're going to take a look at this section now. So that spiral organ, or that organ of Corti, so this section that that I said we're going to take a look at, kind of here it is, more in a blown up view. So we have those three different membranes, the vestibular membrane, the basilar membrane, and this tectorial membrane. This basilar membrane, remember basilar membrane is going to, remember that stratum basale, that basilar membrane basically connects something to something else. So here in this case, this basilar membrane is going to connect that cochlear duct and that scala tympani. It actually forms the floor of that cochlear duct. Here is where we're going to find those hair cells that depolarize in response to sound. So we had hair cells in our olfactory, in our olfaction. We also have hair cells in our hearing as well. And then our tectorial membrane, we're not really going to talk about our tectorial membrane. All right, so our spiral organ, or our organ of Corti, that is going to be our main organ for the sense of hearing. It sits on the upper surface of the basilar membrane. So remember, here is our basilar membrane. So our organ of Corti sits on top of that. So this is kind of pretty much everything else. That contains our hearing receptor cells. They're called hair cells. They also have stereovilli or stereocilia. It's kind of like a bunch of microvilli. And basically those sound vibrations cause that stereovilli to contract and bend against that tectorial membrane. So remember, we're talking about a fluid-filled pocket here. Now, when those hair cells are stimulated, they're going to release a neurotransmitter called glutamate. We talked about acetylcholine being released for skeletal muscle contraction. We talked about norepinephrine in the sympathetic nervous system. Now we're learning about glutamate being the neurotransmitter for hearing. And then we have different frequencies of vibration, so different pitches. So remember that we had that snail spiral shape, and once we unwind it, different frequencies are actually heard on different parts of that spiral shaped organ. And then those nerve impulses are going to go to the cochlear nerve. All right, and this is just a microscopic view of those hair cells on that basilar membrane. So I said that different pitches or frequencies are heard in different areas, and this is just showing that. All right, so remember that picture, that slide that we talked about with the apple, way at the beginning of the lecture. So within that, information is processed within the central nervous system. portion, this is how your hearing gets processed in that central nervous system. So remember, we have that vestibulocochlear nerve. Remember, the vestibulocochlear nerve is cranial nerve number 8. From there, it's going to go to the medulla oblongata, then the midbrain, then the thalamus, then to the auditory cortex of that temporal lobe. Your ears are right next to your temporal lobe. All right, here is a summary slide of kind of what we've talked about so far. You should probably know this slide. Oops. All right, so for hearing loss, about 8% of the population has some sort of hearing loss. We can classify hearing loss as conductive hearing loss or sensory neural hearing loss, or conductive deafness or sensory neural deafness. Conductive deafness, basically, it's the most common cause of hearing loss. And basically this is when we have hearing loss or a malfunction with vibrations being transmitted to the inner ear because of earwax, injury of the tympanic membrane, fusion of the auditory ossicles, that malleus incus stapes, or injury of those malleus incus stapes, otosclerosis, which is basically hardening of those bones, those 3 bones. Sensory neural deafness, here the key word is neural, so neural nerve, so think of damage to the cochlea. Auditory, that vestibulocochlear nerve, those nerve pathways can be caused by long-term exposure to loud noises, factory noise, loud music, explosions, IEDs, brain tumors, brain damage, stroke, use of certain illicit drugs or medications. All right, Jared has been an avid a fan of rock music for many years, has attended numerous concerts, getting close to the stage as possible. He's never worn earplugs or protective wear. Now in his 50s, he has become hard of hearing and can no longer detect quiet sounds or low talking. How might his symptoms be explained? He has developed a brain tumor that puts pressure on his vestibulocochlear nerve. No, nowhere in the question does it say brain tumor. He must have used antibiotic. No, nowhere does it say medications. His tympanic membrane has hardened. Nowhere does it say otosclerosis or tympanic membrane damage. His hair cells are permanently damaged and desensitized to loud music. Yes. So, loud noises will damage your hair cells. So, you don't see the damage of listening to loud music immediately. It's going to take decades for that damage to be done. Just like smoking doesn't cause immediate damage, or vaping doesn't cause immediate damage. You're not going to see the effects of listening to loud music for a couple decades. All right, so with we talked about sensory neural deafness and conductive deafness. So we can treat sensory neural deafness by getting a cochlear implant. Basically, these are not hearing aids. Hearing aids would kind of help with conductive deafness, but a cochlear implant actually helps with stimulating that auditory nerve. Remember, the auditory nerve is also called the vestibulocochlear nerve. It stimulates that nerve. and helps a person hear sounds. And it's actually used typically if your nerve is damaged. Um, Outside of like an explosion or loud, if we go back to here, factory noise, loud music, explosions, sometimes this is present at birth. And if it's present at birth, you know, you can hopefully know that before the age of 3, and we can get the cochlear implant implanted before the age of 3. And that's good because that's when a child is developing their speech and hearing. And if we can get that implanted before they are rapidly developing their speech and speech, then it will just help their growth or speech patterns. All right, so I mentioned that the ear does 2 types of equilibrium, static equilibrium and dynamic equilibrium. So static equilibrium is basically what is your body doing if your head is still. And it says that these receptors are found in the vestibule of the inner ear. And we'll talk about these vestibule things. Dynamic equilibrium is basically what is your body doing, rotation, direction of movement, and movement of the head and body. And it says found in semicircular canals. So here, with static equilibrium, we can think of this as a 2-dimensional world, whereas dynamic equilibrium is more of a 3-dimensional world. And we'll look at, like I said, we'll look at pictures later on. So static equilibrium. Organs are located in these things called vestibules, which is between the cochlea and the semicircular canals. We have 2 vestibules called the utricle and the saccule. And really that's all. We're not going to worry about this. So static equilibrium, vestibules called utricle and saccule. So those are going to be, so this is our cochlea, that's going to be our hearing. We have our vestibules here. We have a utricle and a saccule. And then when we talk about dynamic equilibrium, so remember this is our 2-dimensional world. And then our semicircular canals here, this is going to be our 3-dimensional world. So static equilibrium, dynamic equilibrium. So dynamic equilibrium, 3 semicircular canals, and these canals sit at right angles to each other, so that's going to help us with a 3-dimensional world. So they all sit at right angles to each other. That's a right angle, that's a right angle, that's a right angle. All right, so your head is in motion. What's your body doing? All right, so for vision, vision obviously is the eye. We have some accessory structures. Our eyelids, we actually call those palpebrae. Basically, they're there for protection. Eyelashes are there for protection. We have a lacrimal apparatus. That's our lacrimal gland. Produces tears. Our extrinsic eye muscles produce eye movement. We actually have 6 eye muscles that we'll talk about. Um, our eyelids actually have 4 layers. Um, we have skin. It's actually the thinnest skin in our body for our eyelids. We have muscles that are associated with our eyelids. The orbicularis oculi actually closes our eyelids. Our levator palpebrae superioris opens our eyelids. We also have connective tissue, and then we have conjunctiva, which is a mucous membrane that lines our eyelid. So I said that we have our lacrimal gland that produces and secretes tears. Actually, in our lacrimal gland, there's some antibiotic properties to our lacrimal gland. It contains this enzyme called a lysozyme. So we produce tears that will go into our eye, and they will actually drain into these canaliculi, the superior and inferior canaliculi into this lacrimal sac, and that's why your nose runs while when you're crying, because you are draining your tears. All right, so our 6 eye muscles. There's 4 that are easy to remember. So as long as you know your definition, your 4 rectus muscles. Superior means above, so your superior rectus muscle rotates your eye up. Inferior rectus, down. Medial rectus muscle, rotate your eye medially. Lateral rectus muscle, rotate your eye laterally. So those are your 4 rectus muscles. Now your oblique muscles do the opposite of what they say that they're going to do. So your superior oblique, superior means above, and oblique kind of means diagonally. So you would think that your superior oblique muscle would move your eye above and diagonally, but it doesn't. Your superior oblique rotates your eye down and laterally. Your inferior oblique muscle would actually rotate your eye up and laterally. Now, if you have ever seen someone with a lazy eye, that's usually a problem with your inferior rectus muscle. Typically, you're going to wear an eye patch over your good eye and let your lazy eye work on building up the strength of that inferior rectus muscle. All right, with your eye structure, we have 3 different types, or 3 different kind of layers of the eye. We have an outer layer, a middle layer, and an inner layer. That outer layer is a fibrous layer. Your middle layer is a vascular layer, lots of blood vessels, and your inner layer has a bunch of nervous tissue. This word tunic actually just means protective or supportive covering that encases some organism's part of its body. That's just what the word tunic means. All right, so portions of the outer tunic. We can see that there's the cornea and sclera. So this cornea is what the eye, or what light is going to hit first. And then we have this sclera, which is kind of like this interior portion right here. So cornea and sclera. So the cornea is this outer layer, sclera is more of this inner layer. So the sclera is actually the whites of your eye, and the cornea is like this little film over the whites of your eye. Your cornea helps focus the light rays. It's going to transmit, refract light. The middle portion of your eye, it's going to have this thing called a choroid coat. Remember that middle portion is very vascular, so it has a lot of blood supply, has a lot of melanocytes, which are going to absorb a lot of light. We have these things called ciliary bodies, which are very thick, pigmented. It also changes the lens for refocusing. That lens is what's going to kind of alter the light, or alter those light rays coming into your eye to help focus the light into the back of your eye. We also have the iris. That's the pigmented portion. So you have blue eyes, you have brown eyes. It's anterior to the ciliary body, so it's in front of the lens. It helps control the amount of eye light entering the eye. So here you can see this this ciliary body right here. All right, so we have this also this anterior cavity. This is still portion in the middle portion of the eye. So we have an anterior cavity, which is going to make up this front third of the eye. It's going to be between the cornea and the lens. It's filled with this watery fluid. We call this aqueous humor. So you could actually stick a needle in your eye and actually drain out that watery fluid. I don't recommend you doing that. But basically it's a watery fluid. The lens is going to be transparent. It almost looks like your glasses if you wear glasses. It says it's biconvex. We'll look at that structure later on. Actually, it actually looks like this. That's what your lens looks like. It lies behind the iris. It has a lot of elastic connective tissue to it because it needs to change shape when the light passes through it. It helps us, it's held in place by these things called suspensory ligaments. There's your lens there. And it's going to help focus those light rays onto your retina as light passes through your eye. And I said that the lens helps that the lens changes shape. When it does that, that is called accommodation. Accommodation is when that lens changes shape to view, here it says to view objects. Or to view close objects. So that lens can become thick or it can become thin. It can alter its shape if those ciliary muscles contract. That lens becomes thick. If those ciliary muscles relax, that lens will become thin. So if we're trying to look at closer objects, that lens will become thick. If we're trying to look at objects far away, that lens will become flat. And that just allows, like I said, the light rays to focus on the back of our retina. So the lens will change shape depending on if we're looking at stuff close up or far away. And that's called accommodation. Now, iris. Iris, that is your eye color. And that's due to the amount of melanocytes that produces melanin. Your pupil, that's the black part of your eye. That's the the window to your soul. It's the opening in the center of your iris. It's the size of your pupil. It's because of smooth muscle tissue. It's controlled by your parasympathetic nervous system. We have these radial muscles that are going to contract and relax, that will dilate your pupil or constrict your pupil. If your radial muscles contract, your pupil will dilate. If your radial muscles constrict, if your radial muscles dilate, then your pupil will constrict. All right, so while working in the lab without protective glasses, a research assistant accidentally sprays themselves with a nerve agent that blocks contraction in smooth muscles. How will the eyes be affected? Inability to focus or adjust, or to adjust pupils, maybe. Loss of pigment in the iris or choroid. So loss of pigment that's because of melanocytes. No. Only able to detect black, white, and gray tones. So that's rods and cones. No. That's more genetic makeup. No. Inability to move eyes up and down. So smooth muscle, inability to move eyes up and down. That's skeletal muscle. All right, inability to focus or adjust pupils. You can't control whether or not your eyes dilate. That's going to be smooth muscle. So if we go back up here, and I purposely didn't say this, sympathetic, parasympathetic. That is our autonomic nervous system. That's smooth muscle, adjusting pupils. All right, aqueous humor. Remember, aqueous humor is that fluid-filled chamber, or that watery fluid in our anterior chamber. Don't stick a needle in your eye. It's going to circulate throughout the pupil. It's secreted by these ciliary bodies. Basically provides a bunch of nutrients. And then we have a posterior cavity that contains this vitreous humor. So the anterior contains aqueous humor, which is watery. The posterior cavity contains vitreous humor. Vitreous humor is more thick. It's a gel-like fluid. So, you know, if you stick the needle in your eye and you stick the needle really far in your eye, you're going to squeeze out some jelly. Again, don't do it. So, we have a lot of collagen fibers kind of more in the back of your eye. So we have what's called a vitreous body. Vitreous body is that vitreous humor, that gel-like fluid, plus a bunch of collagen fibers. So now we're going to talk about the back part of your eye, a bunch of that nervous tissue, or that inner tunic. We have what's called the retina, and that's where all of our light goes, or is projected into the back of our eye. It has a lot of visual receptors. Remember, those are called photoreceptors. Has a lot of blood vessels. You see the blood vessels in the pictures. It has that optic nerve. Remember, that optic nerve has to take our vision somewhere to our brain. But we have this spot called the fovea centralis. And it says it's in the center of the macula lutea, and it produces the sharpest vision. So here you can see that fovea centralis. So that fovea centralis basically is, if you're looking straight on at something, the light that is coming into your eyes will project right onto your fovea centralis. So if I'm looking at if I see something in my periphery, that is not projecting onto my fovea centralis. I don't have that sharp vision in my periphery. I have that sharp vision if I'm looking directly at something. So, so whatever is projecting into my fovea centralis, it's because that is what I am looking at directly. Now we also have this thing called a blind spot, or your optic disc, and that's because we have a bunch of blood vessels running into our eye. And because of those blood vessels running into the back of our eye, we don't have any photoreceptors in that area, and that's our blind spot. All right, we're not going to really get into the structure of the photoreceptors, but they're going to kind of resemble the other types of receptors in the fact that they're all bipolar cells. They're going to go to some nerve, in this case the optic nerve, cranial nerve number 1. They're still horizontal cells. And really that's all we're going to talk about. They go to these photoreceptors. Now we have rods and cones, which we'll talk about here in a little bit. So you're going to need to know a little bit about this slide, the outer layer, middle layer, and inner layer. All right, so what is the correct order of structures through which light passes as it travels through to the retina? All right, so remember that the first thing that it touches is the cornea, and the last thing that it touches would be the retina. So we'll do cornea here, and we'll do retina here. And then remember that retina, remember we have that vitreous humor here. Let's look back up here. Well, we can do it here. So we have our cornea, aqueous humor, then let's see if we can find some other structures. So we have our cornea, aqueous humor, then we have our lens, and then we have our retina or vitreous humor. Let's see. Okay, so cornea, lens, aqueous humor, so that one's out. We know our cornea is first, so that one's out. Cornea first, that one's out. Cornea first, that one's out. Okay, so cornea, aqueous humor, lens, vitreous humor. So that one's going to be our answer. All right, so refraction. Refraction happens because light passes at an angle between mediums of different densities. So all that means is as light passes through your lens, light rays are going to be bent. So we already know that your lens changes shape, and that is accommodation. But light rays are going to be bent because we have air here, and then we have your lens, which is going to be a different density than air. Now, the example of this, you can see the little glass right here, and the light rays are bending. But if you've ever, stuck a pole into a pool, if you're scraping the bottom of the pool, you know how when the pole goes in the pool, it actually kind of looks bent. If it goes in the pool, that's what I'm talking about. The pole looks bent because the air and the water have two different densities. That's what's happening. It looks like the pole is bending, but it's actually not. Light rays actually bend because the lens has a different density than the air that the light is traveling in. So the lens will change shape, accommodation, and the light rays will actually bend because of different densities between the air and the lens densities. And that's refraction. So the lens has two functions: accommodation, the lens changes shape, and refraction, the bending of the light rays. Now, because of the shape of the lens, the light rays can refract different ways. Now, convex lenses and this also happens in the cornea as well. So not only the lens, but the cornea will cause the light rays to converge. Concave lenses causes the light rays to diverge. So you can see convex versus concave. The way I remember it is that concave, it caves in on itself. Concave caves in on itself. If you have a concave surface, the light will diverge, it will spread out. Convex, the light waves will converge on itself. Remember, we had converging neurons and diverging neurons. Converging neurons, where you had multiple neurons that linked up with one neuron, and the definition was 10 or more neurons connecting with 5 or fewer neurons. Because of refraction, if we had normal healthy eyes, remember that both the cornea and the lens will participate in refraction, actually about 75% in the cornea. Remember, the lens does accommodation and refraction. The cornea will do, the cornea will do refraction. But in normal healthy eyes, the lens and the cornea will refract that light to put those light rays right on that fovea centralis. Now, sometimes, and actually what we see is that because of all the bending and the refraction and the accommodation, the image that's actually projected is actually an upside down image and a flip-flopped image, a left to right flip-flopped image. But our brain knows that, we don't see the world in an upside down and a left to right flipped image, so our brain kind of reverses that upside down and reverses that image from left to right, and we actually see the world correctly. But sometimes our lens and our cornea doesn't actually process the light rays appropriately, and we have to have glasses or contacts. So we have what's called refraction disorders. Myopia is the medical term for nearsightedness. In this case, our eyeball is too long, and what happens is the instead of focusing the light rays right here in your fovea centralis, the lens and cornea will bend the light rays to focus kind of in front of the fovea centralis. And we just use glasses with a concave lens to put the image back on the fovea centralis. That's all we do. With hyperopia or farsightedness, the eyeball is too short. So the lens and cornea actually focuses the object kind of further than where the eyeball would be, and we just use a convex lens to shorten up where the lens and cornea would focus that object and to project it on that fovea centralis. Now we also have this thing called presbyopia, which is actually farsightedness at the age sometime after 45. And I will say, nearsightedness, you can see things near but not far. Farsightedness, you can see things far but not near. Farsightedness, presbyopia, typically you're only going to see that after the age of 45. This is usually these are people that just need readers. Basically due to the loss of elasticity of that lens. Just give them reader glasses. They're typically fine. Astigmatism. Usually is because of a defect in the curvature of that cornea and lens, resulting in a blurry vision. Usually these are nighttime defects. A lot of times with astigmatism, you'll see, the stoplight or the street lamps, and you'll see bursts of light coming out of the stoplight. If you have astigmatism, you'll know exactly what I'm talking about. Can be corrected with glasses, though. All right, so rods and cones. Rods and cones are the photoreceptors that lie in the retina that help us see in black and white, color, grayscale, and such. So rods have obviously a rod-shaped appearance, and they're very sensitive to light, more sensitive to light than cones are. So they help us see in dim light. They help us see in shades of gray and the outline of objects. Cones actually produce very sharp images and lets us see in color. Help us see in bright lights. They contain these light-sensitive pigments called iodopsins. We have 3 different types of iodopsins. Erythrolabe is red light, chlorolabe is green light, cyanolabe is blue light. And so by combining, red, green, and blue, we can kind of see all of the colors of the spectrum. Now, if you are colorblind, it's because you have a lack of a type of cone. And typically, colorblind people, they're first going to be not be able to see shades of green and or shades of red. All right, now remember that I said that fovea centralis it produces that sharpest vision. It's what you see straight on. Fovea centralis only contains cones. because you know when you're looking at something straight on, you want to be able to see that the sharpest vision. So you're going to be able to see in color in that fovea centralis. Now, so here's just a picture of what rods and cones look like. Not really going to go into that. So, why are images in your peripheral vision blurry, or that lateral retina blurry? Lateral retina, your peripheral vision, because there are more cones than rods, there are more rods than cones, there are no rods, or because your retina is thinner. All right, so your answer is going to be B. So it's blurry because you're going to have more rods than cones. Remember, cones produce sharp vision. So answer A is going to be out. We're going to have more rods than cones in this region, so answer C is out. The retina is not thinner, so answer B. All right, so when we, you know, you're in a dark room, and you're using your rods. Oh, you're using your rods. You're in a dark room, and then you have what's your rods produce this thing called rhodopsin, and then now someone comes in and flips on the light, and oh my God, it's way too bright. Your eyes have your eyes have a lot of rhodopsin in them, and once those lights come on, your rhodopsin actually decomposes in the presence of light into two chemicals. One's called opsin and the other's called retinol. So rhodopsin decomposes into optin and retinol, and then as that rhodopsin gets depleted, someone now shuts off all the lights and you're in the dark room again. You can't see. It's going to take time for that rhodopsin to build back up. You need to kind of regenerate that rhodopsin back in your rods in order to see in that dark room again. All right, we've already talked about these three colors. Just make sure you know that you should be able to remember erythro. Remember, we talked about erythrocytes. Those are red blood cells. Cyan means blue. Chloro means green. Chlorophyll. You've talked about chlorophyll in probably Biology 1000. All right. Arnold enters a darkened movie theater on a sunny afternoon. At first, he can't see objects. After several minutes, he can see more clearly. What happened? The optic nerve has fatigued. No. We just talked about it. Iodopsins, which are depleted in sunlight, have to rebuild. Okay, we'll keep that answer for now. The iris, which needs time to correctly adjust. No, that would happen automatically. Rhodopsin, which was depleted, has to rebuild. So now we're left between rhodopsin and iodopsins. So the answer is going to be D, because rhodopsin, remember, gets depleted into opsin and retinol. So here, iodopsins, those are what helps us see color. So that's why B is going to be out. All right, stereoscopic vision. That's what lets us see in 3D. So if you're seeing something up close, you know, you cover your right eye, you cover your left eye, cover right eye, you can see like kind of two different images. And what happens is your brain actually sees two different images, one from your right eye, one from your left eye, but then your brain actually merges those two images so that you can actually see a three-dimensional view of that image in a three-dimensional world. That's just what stereoscopic vision means. Now, going back to that chart that we saw with the apple and that visual process and that processing thing, you know, integration in that central nervous system, from a inside the central nervous system, what happens. You know, we know that we go from the retina to the optic nerve. The optic nerve is cranial nerve number 2. Then where does it go from there? Well, we know that vision is processed in that occipital lobe of the cerebrum. So that optic nerve goes through the optic chiasm, through the optic tract, through the thalamus, through these things called the optic radiations, through the visual cortex. We did this in lab, actually. You did a lab on this. It'll be actually the very last lab that we do, special senses lab. All right. Remember that the left side of the brain controls right side of the body and everything. So that we had that crossover. We'll see that crossover on the next slide. So here we have the eye. And depending on what part of the eye that we're talking about, we're going to see that crossover in different sections. So if we're talking about the medial aspect of the eye, that medial aspect of the eye, that medial fibers from the medial side is going to cross over at that optic chiasma. So that's why we had that statement here. Fibers from the medial side of each retina cross over in that optic chiasma. All right, age-related changes. Remember, it sucks getting old. Damage to hair cells in the spiral organ. Loud noises. You might need hearing aids. Tinnitus. Tinnitus is just ringing in the ears. Age-related smell and taste problems. You're going to lose your olfactory receptors. What you could smell, you might not be able to smell anymore. That's called anosmia. Age-related visual problems, dry eyes, floaters, loss of elasticity of the lens. You might need glasses, you might need readers. We talked about presbyopia. You might need that readers as you get older. Glaucoma. Glaucoma is basically you have increased pressure in that anterior chamber. That watery fluid, that because you have excess aqueous humor. You might have cataracts. Basically, you just remove the lens in a cataract. You might have macular degeneration. So that is it for A&P1. So please let me know if you have any questions. Otherwise, hopefully I will see you in A&P2, or in another class, or somewhere else around. All right, take care. Bye.